nerves to contract your muscles. Okay, let's talk about the nervous system. If you haven't picked up something about how I do my presentations, on the outline slides, I put one of the most key figures on the outline slide so that you see it like multiple times. Wait, someone just said new Texas head coach. Who's the new Texas head coach? Oh, Steve Sark. Yeah, Sark. Okay, anyway, I'm way distracted today. All right, so in terms of what you should take away from this lesson, when you're studying, if it's on the outline slide, you would better be able to explain exactly what it means. Be able to draw it and explain how it's going to change with exercise training, how it's going to change with age, lots of different things. Just if it's on the outline slide, definitely know it. If it's on my slides, know it. And if it's on the outline slide, you better know it really, really well. So we'll get into what this diagram is a little bit later. So how do muscles generate force? Lucky for you guys, we filmed me the other day. I have to go share my screen in a slightly different way, but I'm gonna play a video of me to help introduce this topic. All right, so TAs, if you can't hear it well, just let me know. I can't see any of you people. My Brady Bunch screen goes away when I share in this optimized way. All right, today we're going to talk about how action potentials elicit muscle contraction.
We can't see anything.
When I do a bicep curl,
thank you,
the muscle moves. All right, redo. Optimized. Thank you, Abigail. All right, let's try this again. Let me know if you can see and hear. All right, today we're going to talk about how action potentials elicit muscle contraction. As you can see, when I do a bicep curl, if you go to the chat, okay, looks and sounds good. All right, good. The muscle moves, right? What's happening? I'm sending electrical signals from my brain down to the muscle to cause the muscle to shorten. And then I stop sending signals and it relaxes and everything falls down. So it's all about electricity going from my brain down to the muscle. What's interesting is that we can hijack this system and completely bypass the brain. Today I'll show you something called an electrical stimulator. You might have seen this in a physical therapy clinic, which will send action potentials from the stimulator directly into the muscle without me even thinking about it. This is an electrical stimulator. What's going on is that it's sending an electrical current out of this little device that has kind of a battery in there. The electricity is being sent down the cord, going from one electrode to the other. As it does that, it's passing through my muscle, and you can see that it's causing a little bit of a contraction. So what is it about the electricity that causes the muscle to contract? I can tell you it's not my brain. My brain is not thinking about doing this right now. It's happening completely on its own. So there's something in the muscle that senses this electricity and causes contraction. What is it? So to help you understand how action potentials cause muscle contraction, I'm going to show you two different electrical stimulation patterns. Now this first pattern is going to be a constant stimulation. So it'll send an electrical current or electrical stimulus. It almost feels like a pinprick, and it will be rapidly followed by another stimulus, one right after the other, almost like a drum beat, like ta 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 ta. Okay, so watch what happens when I stimulate it hard enough. Can you see this? I'm not making this happen. So it's relaxing now, and now I have a stimulus causing the muscle to contract, and now relaxation. So it's causing, it's a little painful, so don't do this at home. But what is it? What's causing the contraction, and why is it causing it to flex? If you notice, you can see my hand also flexing. So not only is the bicep causing contraction, but I'm also getting some of the hand flexing. And what's going on there is that some of the electricity is passing through the nerves that innervate these muscles down here, and it causes it to contract. So even just electrically stimulating the nerve can cause muscle contraction. Most of it's stimulating the bicep, which is causing this bicep curl. All right, so let me reshare my screen, not optimized here. So how many of you guys have ever done electrical stimulation on yourself or like in a PT clinic? All right, so a lot of you guys. How many of you, it's just electricity, right? So if you haven't done electrical stimulation, how many of you guys have licked a 9-volt battery? I know you're out there. I know you've done it, okay? I tried to get my 7-year-old to do it the other day. He wouldn't. So he's a little smarter than I thought he was. So I guess that's good. So what we're doing, it's electrical stimulation. Electricity, that's all it is. And in this case, I'm applying it directly to the muscle. So, all right, so come on. All right, so I'm applying the electricity directly to the muscle with a little bit of residual getting into the nerve. Now the question is, how is electricity causing my muscle to contract? So let's go through, I'll walk you through some of the main ways of muscle contraction. Hopefully this is review. It's okay if you've kind of forgotten it, but it should sound at least a little familiar. And you'll read more about this in the textbook. So your muscle contraction happens with lots of tiny little molecules, actin and myosin, interacting. So the myosin is this part here, showing a lot of different orientations. It looks almost like a hammerhead. And so the myosin will reach out and latch onto the actin and pull it. So here, the myosin is not connected to actin. This is actin, a small little, almost looks like a strand of pearls. So when the myosin reaches out, it will contract and pull the actin towards it. And when that happens, we get shortening, okay? So the actin and the myosin grabbing will cause shortening. Get ready for the animation of the year. So we got, on a different way to look at this, the actin and the myosin overlap. And when you do a full contraction, you get more and more overlap. So here's the animation. Myosin pulls the actin in towards itself, in towards the center. Ready? So it's the myosin pulling the actin in. That's a contraction. That's how we generate our force. And the movement of that myosin requires ATP, and some calcium comes into play. But this is at the most fundamental. Actin and myosin interacting causes the contraction. So let's take it a step further. So how does an action potential coming from a nerve actually cause a muscle contraction? So in what might be the second best animation of the year, you should remember this from the other day, an action potential travels down the axon. When it hits the end of the axon, it causes a release of acetylcholine that goes across the neuromuscular junction, binds onto a receptor, which then allows a little bit of sodium to come in. As the sodium levels increase little by little, these are EPSPs, essentially. When one acetylcholine binds onto the receptor of the muscle, the membrane potential or the voltage goes up a little bit towards threshold, not all the way up. It's just a tiny graded potential, or almost an EPSP in concept. So once we get enough acetylcholine binding and enough sodium going in, we generate an action potential. And that action potential is just sodium channels opening up so that a lot of sodium flows into the cell. Just like in the neuron, the exact same process going on in the muscle cell as in the neuron. We have sodium flowing in, potassium flowing out, and then you have the sodium potassium pump restoring all the gradients, same things, okay? So now, how does an action potential lead to muscle contraction? First here, let's look a little more closely at what goes on in this space here. Okay, so this is called the neuromuscular junction, the space between the neuron and the muscle. Pretty simple. So when the action potential travels down, you're changing the voltage right here at the end. And you have a channel, a calcium channel that's sensitive to the voltage. So as soon as the voltage flips with the action potential, calcium goes in to the neuron, and that influx of calcium through a series of steps will cause acetylcholine to be kicked out. Now, acetylcholine goes out into the junction, into the neuromuscular junction. And in order for it to do anything, it has to bind to the receptor. So you have these receptors on the outside of the muscle. So the acetylcholine goes and binds onto here. So it looks something like this. And when it binds on to that receptor, a little bit of sodium goes in. One acetylcholine is not enough to get the job done, right? One acetylcholine does not guarantee an action potential on the muscle. Just like one EPSP on the neuron does not guarantee an action potential, same thing on the muscle. You need multiple. You need a lot of acetylcholine released in order to get the action potential generated. Once you do get enough sodium in from the acetylcholine receptor, it will stimulate the voltage-sensitive channel, voltage-sensitive sodium channel, which will then allow a ton of sodium to come in, just like in your neuron. Okay, so this completely review. The only thing that's different is we're looking at a different cell. Instead of these channels being embedded in a neuron, now they're embedded in a muscle. All right, so now how does an action potential or a flipping of voltage, a bunch of sodium flowing in, how does that cause actin and myosin to interact? Well, the link between actin and myosin, the action potential in actin and myosin is calcium. Calcium is basically the secret ingredient here. Without calcium, any action potential you generate will not cause muscle contraction. So as the action potential, oh, is there a question? No. As the action potential travels down the membrane, we have a wave of depolarization, just like what happens on the nerve. It's going to stimulate these channels, DHP and RYR. What that means is dihydropyridine and ryanodine receptors. These are two molecules that the DHP are sensitive to voltage. And when the voltage stimulates them, they kind of move. And it basically pulls a cork. The ryanodine receptor is like a cork holding all the calcium inside the sarcoplasmic reticulum. Okay. So at rest, right now, if your muscles aren't contracting, most of your calcium is going to be stuffed into the sarcoplasmic reticulum. It's basically just a giant balloon full of calcium. Once the action potential stimulates these DHP receptors, the ryanodine receptor or the plug in the balloon or the SR opens up and calcium floods out. So action potential causes calcium to flow out of the SR, sarcoplasmic reticulum. Okay, we've gone a while without a pause. Questions?
One more time what the cork was.
Yeah, so the cork essentially is the ryanodine receptor. So we have a ton of calcium stuffed into this SR. And if we just open up a hole in the membrane here, it will naturally flow out. And so let's just say like right here, this would be the ryanodine receptor. And if we pull out the ryanodine receptor, all the calcium goes out that way. So we just move that and calcium can flow out. You'll get more detail as you read in chapter 8 about this. Right now, I'm just kind of getting you the generals.
There were a few questions in the chat that just came through if you want to look at those really quick.
Okay. So question was, how many of these terms do we need to have memorized in terms of ryanodine receptors, circupumps? If it makes it onto my diagram, you better have it memorized. These diagrams that are obviously like homemade, they are so simplified down to like the core essentials. So if it makes it on there, makes it on one of my diagrams, make sure you have it memorized. That brings up another really good point. When you're studying for this test, which is in about 2 weeks, if you cannot draw exactly what's going on from the nerve to muscle contraction, how sodium moves, how calcium moves, how the actin and myosin interact, if you can't draw that out and explain everything, you're not going to do well on the test. That is basically the way I designed this test. Is I went through and I wrote on my board over here, I drew out all the steps from action potential in the neuron to muscle contraction and relaxation in the muscle. Once I drew it out, then I asked myself, okay, what happens if I take this out? Or what happens if I take that out? And that's how I designed the questions. So if you understand this general concept, you'll be able to apply and answer any question I throw at you. If you can't explain how muscles contract, there's a really good chance you'll fail this test. So please study up. So then there's a question, where does the calcium interact with actin and myosin? So that's what comes next. So how does calcium actually cause a contraction? So I'd already shown you this kind of deal. Let me go back. This showing that the myosin and the actin interact. Well, at rest, you got something that prevents them from interacting. And what that is a troponin and tropomyosin complex here. So on top of the actin, you have, this would be troponin. They're little tiny molecules where calcium will bind. The troponin is attached to what almost looks like a giant rope, in my opinion, to something called tropomyosin. And you can see in the word, tropo referring to troponin, myosin referring to the myosin head, right? So what the tropomyosin does, it blocks the binding site for myosin on actin. So myosin wants to bind right here, but it can't because you have the troponin blocking that. Now here's where calcium comes into play. When calcium comes in, is released from the SR, the calcium will make its way to the troponin and it binds onto the troponin. And that causes the troponin to change its shape. And as it changes shape, it moves and moves the tropomyosin away from the binding site. Okay, so calcium binds onto troponin, which causes tropomyosin to move. And now with that exposed, now the myosin can interact with the actin. So without calcium, you're not going to get any force generation. So now let's do kind of a little quiz here. So watch my arm. I'm going to use my left arm to move my right. My right is not doing anything active. It's moving back and forth because my left arm is pulling it. So when my arm is moving back and forth like this, or being passively moved, do I have actin and myosin interacting? I don't. The calcium hasn't been released, and so the actin and myosin just slide past each other without interacting. They don't get to hook up. When I actively contract like this, now I'm releasing calcium, and actin and myosin can interact and hook up. All right. Questions at this point? So then, okay.
I just have a really quick question. Just about this recording. Are you going to post this recording so we can come back to it for reference and study it again?
Yes. So I should have all the recordings already posted. If you go on to Learning Suite, to the BYU Online or the Online tab, There's like cloud recordings in there. You should be able to find previous classes.
Perfect. And then are these slides also, like, are these on the content tab? Or?
Yeah, so I posted these slides maybe 3 minutes before class.
Okay.
I was working right to the last minute to refine this.
Yeah, the animations are incredible.
They're on point, right? Yeah, you go for a PhD, they don't teach you these things. They don't teach you how to make animations. They also don't teach you how to teach, which probably shows sometimes. It's a complete tangent, but I'm sure you've noticed with a lot of your teachers, you can be like the world's smartest physiologist or chemist, but not be able to communicate an idea. So that's one of the holes in the education system. Professors are never taught how to be teachers. Okay, so, oh, someone else, yeah, post-secondary teaching. Yeah, we'll have to talk sometime, Dylan. So, okay, here's a great question. Actually, first, before I get to the great question, let me go to a almost great question. So how does the contraction stop? If I release the calcium and it just stays in there, the tropomyosin will stay away from myosin and you'd keep a contraction. So the key to stopping your contraction is getting the calcium back into the SR, getting it away from the troponin, and so you can close off actin and myosin from each other. So then you have these Circa pumps. Let me go back. So when you're releasing calcium from the SR, it just goes out naturally because it's stuffed in there. You have a huge gradient where you have a lot of calcium in the SR already. So as soon as you open the door, the ryanodine receptor door, the calcium flows out without requiring any energy. But when you want to stop the contraction, you need to shove the calcium back in. And that's where this Circa pump comes in. So Circa is going to be like the word you can use on a date to impress someone, or like when you're talking with your mom or dad or friend, and they ask you, what have you learned in college? You can tell them Circa stands for sarcoendoplasmic reticulum calcium ATPase. Or just Circa. Now you understand why we just call it Circa. So if you dissect what I just said, it's just a pump that takes calcium back into the SR. And the A on here stands for ATPase because it requires energy or requires ATP. So in order to terminate your contraction, you have to take the calcium back into the SR. And that requires ATP or energy to do that because you're going against a gradient. Now, here comes a question. So I showed you a video of electrical stimulation. Let's say you're working in a physical therapy clinic or wherever, and you have a patient come in and they have a spinal cord injury. They're paralyzed from, let's say, from the neck down, right? So they broke their spinal cord right here. They cannot voluntarily contract those muscles. They can't walk, they can't use their arms. Can you get the muscle to contract with electrical stimulation? What do you think? You're all just looking at each other's Brady Bunch picture and seeing it go up and down. Okay, you can. If you place the electrical stimulator on the muscle, it will contract. What if I place the electrical stimulator on the spinal cord right here? It won't. Well, shoot, that's below the break, it might. Let's go to this guy. Let's say the break is right here. This is a little easier to illustrate. So if I place the electrical stimulator here, I can't get the leg to contract because there's a break right there where the spinal cord is no longer continuous. The nerve is not connected. So you can use electrical stimulation on a person who's paralyzed if you put it directly on the muscle. You can even use electrical stimulation on a muscle that's taken out of a person. So in our research in our lab, With Dr. Hildahl, we do a lot of muscle biopsies where we take muscle samples out of people's quadriceps, just little tiny things. But if you take that muscle sample and stimulate it with electricity and look at it under a microscope, you can see it get smaller. So without any nerves, you can still get the muscle to get smaller. This is an MRI from a study that my friend did a while ago, where he was comparing artery health between healthy controls and people that had spinal cord injuries for like 6 or 16 years. So this is the leg of a healthy control that can walk around. That's the quadricep right there. Okay, so those are the quads. You got your hamstrings and all the other anatomy things that you've had to memorize in the past that you forget as soon as you graduate. Case in point. So we got the quads there. They're nice and big. This is a person after 6 years of being paralyzed, 6 years without using the muscle. Here's someone 16 years. So there's still a little bit of muscle in there. Not nearly as much as there used to be, right? So you can get it to contract. If you can get the electrical signal to get down to that little muscle, you can make it move. But without the voluntary use of that muscle, you get atrophy, major atrophy, and then infiltration of fat. Okay. I put these pictures on almost just as, pretty much just as a tangent, but they're really cool pictures. Okay, so Haley has a good question. I was hoping someone would ask it. So will electrical stimulation alone prevent atrophy if it's used frequently enough? So there have been a lot of studies. The way we study this in research is we either put someone in a bed for several days, like 5-10 days, and don't let them get up. And when we do that, their muscles shrink by about 15%, or we put them in a cast. And over 2 weeks, the muscle shrinks by about 10-12%. So if you electrically stimulated muscle that it can't be used, you can minimize the atrophy. Atrophy will still occur, but it won't get as small. So there's a lot of utility to it if you're trying to keep the muscles healthy, and especially the arteries healthy. Okay. Now, let's talk about what mechanisms your nervous system uses to adjust how much force you generate. And the two mechanisms are going to be very similar to temporal summation and spatial summation. Not exactly the same, but conceptually very similar. Are there any side effects to using too much eStim? I don't know. Well, actually I do. So when I was filming this video, and you'll see in a second, when I was filming this video, my hand hurt for like the rest of the day. I had it in this video, I had it way higher than you would normally put it on in a physical therapy clinic. So it can be painful. My hand was tingly. You might get burns on there. So you have to be careful. The other question that I know somebody is thinking, and we'll talk about this more later, but to use electrical stimulation to get muscle hypertrophy or growth of muscle, not super effective. So don't plan on just going to bed and stimulating your muscles to get them swole. There are some things you might be able to do, but we'll talk about that on another day. So how does your brain send the signal to tell your bicep that you're curling 10 pounds and not 100 pounds, or vice versa, right? How do you differentiate that? How do you get your muscle to generate 10 pounds of force and 100 pounds of force? It's an interesting question, and we're going to start looking at it with this video of this guy whose shirt looks really funny in these videos. Apparently, you'll see what I mean. So let me change how I'm sharing this to optimize it for screen share or for video share. And so what I want you to see, remember how we did the stimulation before. It was a constant stimulation, what you call a train. And So when on that first stimulation, it was going zap, 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 zap really fast. Now I'm going to do a different one, and you'll see how it's different, how the result is different. So in the second stimulus pattern, it's more like a twitch. So it's going to send out a single electrical stimulus and wait about 3 or 4 seconds, then it'll send out another one. So there's a lot of time between the stimuli. So here you go, watch what happens. I'm gonna put it to the same intensity it was at before. You can see it's causing the bicep to contract. It's getting a twitch, but it's not causing me to flex. Remember, the last one would cause me to go all the way up and down. This one's not doing it. The electrical stimulus is exactly the same in terms of the intensity, right? So the question is, let me rephrase that. The electrical stimulus is the same. It's reaching the same number of muscles, right? It's just as intense. The difference is the frequency. Why is it not causing a full flexion? Why can't I get a full contraction here? Now, the answer might seem a little obvious in this kind of scenario where things are kind of extreme, but the answer to why I'm not getting a full contraction here actually has a lot to do with why your muscles fatigue, why you can't generate as much force as your muscles fatigue. Okay, now that's our last video, so let me change how I'm sharing again so I can see you guys. All right, so the question is, why at a low frequency stimulation, why when I'm just going zap, 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 why don't I get a full flexion? What are your thoughts here? If you have an idea, just go ahead and shout it out.
Wouldn't it be similar to having less of the EPSPs like firing at the same time, having a bunch of different motor units come together?
Okay, so you're saying like EPSPs firing at the same time. So when you say a bunch of different motor units together, what do you mean by that, Allison?
So like the summation idea that every time that it gets an EPSP, it's requiring more power because there's more stimulation. And so it's getting more of the type 2X motor units with fatigue faster.
All right, so you're jumping ahead a little bit. So we have two types of summation. We have spatial summation in the nerve, which is something really similar in concept to what Allison's talking about. That's not how I'm changing the force generation here. I'm actually changing the force generation with the idea of temporal summation, or sometimes you might see it written as rate coding. So when you get, if this is a diagram of force, and here's time, if I get one little action potential to cause a release of calcium in my muscle, force goes up a little bit and comes back down really fast. In a couple of seconds, if I do it again, force goes up and comes back down. And then if I wait another couple of seconds, force goes up and then back down. But if I go more frequently, let me change my color. So if I go more frequently, where I'm not waiting as long, before it has a chance to go all the way back down, I'm adding another action potential and another release of calcium, another actin and myosin going like that. And so I can increase the force and increase the movement. So this first idea of how you can increase how much force is being generated is increase the frequency of stimulation. One little action potential is only enough to get this. Probably not even that. Like when your eye's twitching, that's probably one action potential. Annoying as can be one action potential. If you're trying to move your arm, you need hundreds of action potentials in series to get them to go up. If you want to lift 100 pounds, now you need a lot more, like thousands of action potentials in series to keep getting these things to contract. So the frequency of stimulation will increase force.
I have a question, Professor, about that.
Yeah.
So you're saying that the frequency of stimulation, so the myosin will bring the actin together and then kind of like go apart again and bring it together very quickly, right? Very rapidly.
You got it. Yeah.
So our muscles are technically, they're twitching really, really ridiculously fast, right?
Really fast. Yeah.
And so does that happen? Like, does that signal come with the duration of how long you're holding it? Because imagine, like, so if you're like holding a pencil up, you're only using a few muscles, right? But you're consistently using those muscles the longer you hold it up, right?
Yeah. So you have the frequency has to be enough. If your frequency isn't enough to get a full contraction, you would drop the pencil.
Oh, so the frequency intensifies as the more weight
Mm-hmm.
added then?
Yeah, you got it. So if you're doing like tug of war, the idea of frequency would be if you have like your rope, you're just like going like this, trying to pull the rope. This would be a high frequency, and you're making that rope move really fast. This would be a low frequency, and so you're not going to get as much force. But now the second component, when you're holding that pencil, or when I'm doing this, I'm only activating a few of my muscle fibers or my motor units. I don't need to activate all of them. So the rest of them, there's not calcium being released, and so the actin and myosin are just gliding past each other. So now the second way that we increase force is by increasing motor unit recruitment. Let me stop annotating. All right, I can do this. All right. So our first way to increase force, temporal summation, increasing the frequency of action potentials in a given neuron. Now, the second way is to increase how many neurons and accompanying muscle cells are activated. So you have something called a motor unit. And a motor unit is a neuron, a single neuron, and all of the muscle fibers that it innervates. When I say muscle fiber, that's the same thing as a muscle cell. Now, you can see here's a single muscle fiber, or a neuron coming out of the spinal cord, and it's going down and hooking up with 3 muscle fibers. Every neuron is going to innervate multiple muscle cells or muscle fibers. And how many muscle fibers it innervates will tell you how much force you can get for a given neuron. If you just had a neuron that innervated just one little muscle cell, activating that neuron would give you very little force. But you have these other motor units, other neurons, that are innervating like 1,000 muscle cells all at once. So if you generate an action potential on this neuron, it's going to send an action potential to all of the thousands of muscle cells that it's touching. Now, so this is a motor unit. Another key concept with motor units is that a muscle is only innervated or only touching one neuron. So you can see this muscle fiber is only listening to that neuron, whereas this one is only listening to the other. If you had a muscle fiber responsive to 2 different neurons, you'd get conflicting signals on when to contract or relax, and your movement wouldn't be very good. Okay, so now let's take this a step further. Our motor units vary in size and strength. We can break down the size of our motor units into small, medium, and large. Here I just have small and large. So we got a single muscle motor neuron that's connecting with 3 muscle cells. In reality, if it's like in your leg, it's probably, the smallest motor units are probably connecting with like 300 cells. And the biggest motor units are connecting with like 3,000 cells, okay? So you only have a few cells that are responsive to this one. So if I want to generate just a little bit of force, I'm going to recruit this one. If I want to generate a lot of force, then it would probably be a good idea to recruit that one that activates a lot of muscle fibers to contract at once. So this introduces us to something called the size principle. So hopefully you watched this video before with this diagram. But let's say I want to generate this much force, just that much or that much power. I can get away with just one motor unit doing that. And it'll be this small motor unit. So I'll activate this first small motor unit, it'll generate force, and that's the one I'm going to use. Now, what if I want to generate this much force? Now I have to use, let me change my color. I have to use that first one. So this is the first motor unit, same motor unit all the way across. So I'm using that first motor unit still. Now I have to add on a second one and a third one to get that much force. I'm using the small ones still. You always, almost always, use your small motor units first. Only when you get really desperate do you start to opt for your larger ones. So if I want to generate this much force, let's look over. I'm going to first recruit my 3 small motor units, a 4th medium size, and a 5th and a 6th medium size motor unit. So to generate more force, I'm just recruiting more motor units, and you always recruit these small motor units first. You always have them going on in the background. They're your most efficient, they don't fatigue very much, so they're your best option for default. So let me change out of this. Oh, come on. Using the Zoom annotation tool is awkward sometimes. All right, so notice that the small motor unit, right, this little guy, I've drawn it to where each packet or each square doesn't generate as much force. It's vertically not as tall compared to this one way up here, this type 2X or super fast twitch fiber. So when I activate this big guy, I can get more force. And the reason for that is that there are more muscle fibers responding to that single nerve. As soon as I activate that nerve, it's innervating thousands of muscle fibers instead of just 2. So when I activate that motor unit, I get more force. There's another thing going on that we're going to talk about next class, where it's not just the number of muscle cells that make these large motor units generate more force. If you had a small motor unit and a large motor unit with the same number of cells, the large one would still generate way more force because the muscle is supercharged, basically. And you're going to learn why on Wednesday. All right. So now the question comes up, why would you always, physiologically, why would you recruit the type 1 motor units always before the type 2? So this is, let's say this is my type 1 motor neuron, like looking down the barrel of the neuron. It's smaller than the type 2 motor neurons. The type 2 motor neurons are innervating way more muscles, so they need a lot more neural tissue to reach out to the different muscle fibers. So they're much bigger around. Now let's think about how you generate an action potential to get a contraction. To generate an action potential, you need an EPSP to bring sodium into the cell. And let's just say that in order to get an action potential, I have to fill that motor neuron with sodium. So it's going to take me several sodiums, about, what is that? 5 sodiums to fill up that type 1 motor neuron because it's small. But to fill up that type 2 motor neuron, it's huge. I need a lot more sodium and a lot more EPSPs. So it requires a lot more stimulation on that motor neuron to get it to finally go. So while I can get this one up and running with just 5 small EPSPs, this one's going to require like 25. This is why you recruit the small motor neurons and the small motor units first. It's easier to generate an action potential. It doesn't take as many EPSPs. It doesn't take as much sodium to get it up to threshold.
Hey, Gifford, really quick.
Yeah.
We've had a few questions about the scale of your recruitment graph, and I just want to mention to everyone, it's not to scale, it's just for visual understanding.
Yeah, so this is what you should take from this is the type 1 motor units, or these small motor units, generate less force, and then you have these medium motor units that will generate more, and then you have these type 2X or large motor units, they generate the most. But it's not exactly to scale, but that general trend will hold up.
Hey, Dr. Gifford,
Yes, sir.
what is the effect of blood doping on this size scale? So like, does that just allow someone to use their medium and large muscles a lot longer?
So blood doping, you're bringing more oxygen in so that you can make ATP and fuel, where is it? This part. So the blood doping allows you to have more ATP around so you can keep doing contractions. It's not really going to change your recruitment pattern or the size principle all that much. Okay. Now there is kind of one exception to the size principle I want to get to. Whenever you're doing any kind of exercise, as you increase exercise intensity, you're going to start with your type 1 or small motor unit, and then add on the type 2A. And if that's not enough, then you add on the type 2X as well. So you always have the type 1 going, and then you just keep adding on the other motor units on top of it. That's like 99% of your activities. But in very explosive events, something like a baseball pitch or a power clean that lasts less than a second, through practice and neural learning, you can learn how to send IPSPs, inhibitory potentials, to those small motor units, which are also slower to contract. The actin and myosin are slow. You can learn how to inhibit those so that you can just recruit these big ones that are super fast. All right, that's all we have time for. You feel free to stick on and ask questions if you need. See you guys Wednesday. Make sure you read chapter 8, do the learning path and quiz, and we'll see you Wednesday.
I have a question.
Yeah.
Is it accurate that someone who, say, has a lot of explosive power then goes to a lot of like endurance training, that they could and even though they kept doing the same explosive training, they could lose some of their explosiveness because the small fibers are no longer inhibited? I've heard that, but I don't know if that's actually accurate or not.
The explosiveness,
just because it's a case a lot of people use against like running, like this is why I don't run.
Just the slightest, if at all. You see it more with force. So like there is a little bit of interference between endurance training and strength gains. How they elicit adaptation, they conflict. So if you're trying to get as strong as possible, endurance training can blunt your strength gains by about 7% or something like that. It's not huge. So what I would say though, unless you are competing in the Olympics or on a competitive team, the impact of endurance training on any kind of strength or power gains you might be getting is not anything to worry about. You're going to get more benefit from having that endurance training on top of it for your health and how quickly you can recover. So unless you're like on a team right now, when you're specializing in an event that's less than 5 seconds, you should probably have endurance training going on.
Okay, thank you.
Question.
Yeah.
So what happens when someone's like, for example, like squatting or they're like benching and they fail? Is that just like they can't recruit enough? They can't
Got it.
enough muscle fibers or whatever to push it up. So does that just cause all the myosin and actin to like let go, or?
Well, you have like this like pushing fight, or the forces, your legs are generating force up and the weight is generating a force down. As you're doing, let's say you've done like 5 reps on a really heavy squat and you're on that 5th one going up, your legs start to shake. What's happening is particularly these type 2 muscle fibers are starting to fatigue and they can't generate enough force. And so in order to keep going up, the force from the legs has to be greater than the weight. And now that your type 2 fibers are tired, the force from the weight wins and you fall. So that's what's going on. We'll have, in a couple of days,